question for every one of you. Do you want to give it a shot and guess what that is? I can help you, but does it look more familiar to you now? I can tell you, it's an IgG molecule, and it contains all of the disulfide bridging patterns. And the entire structure here, which you've seen in the first image, was actually drawn by Marvin. The 2D clean of the entire structure ended up in the result you have seen here. It takes about 2 to 3 minutes to calculate this. And this is the kind of condensed representation when you're using the individual amino acid residues as superatom groups. So obviously, classical cheminformatics tools don't work well for the representation of such large molecules as antibodies, antibody-drug conjugates, and things like that. We are looking today at how to potentially enable the registration of large molecules by extending the capabilities of classical cheminformatics and still maintaining compatibility with the classical bioinformatics sequence-based tools, which are still kind of the major workhorse for any sorts of sequence assessment of proteins, antibodies, enzymes, and so forth. Just a quick reminder from yesterday, you may have heard from Effie, who is the product owner of our Marvin Sketch product. This is like an enlarged view. You have seen that we are now able to display these sorts of interchain bridges within peptide chains. Interchain bridges where you can identify bridges between individual amino acid residues or cyclizations in cyclical peptides. When you draw these, we have integrated another feature which lets you somehow drag from free and available attachment points a kind of colored markup towards residues which still have another free supergroup attachment point. And when you drag the bond down, then you will see these kind of clean up nice bond. The same thing works if you are connecting to something which actually has more than just a single free attachment point. You will be shown then a little pop-up window here where you have to specify, okay, to which of the free attachment point is your bond actually going to. And in this case, I choose the cysteine here as the first residue, and I get this closed loop for the cyclic peptide. So it's nice. We can draw such things now a little bit better in our Marvin Sketch application. However, that's just display and visualization. What we really need in the backend is an improved data and information management system which is able to kind of cope with these large molecules. And I would like to somehow elaborate a little bit on how we see such a data information management system, which in the core largely is a registration system comprised of a variety of modules dealing with individual parts of a typical workflow of a biotherapeutical development. So we have individual modules, small and kind of atomic, elemental, dealing with the registration of small molecules on one side. We are looking at the registration of macromolecules, such as proteins, small inhibitory RNA, nucleic acids, any sorts of things like that. We are looking at registering biomaterials. This is like cell lines, for example, which are in the end producing your macromolecules. This can be any sorts of molecular biology tools, vectors, plasmids, everything which is needed in order to create your macromolecules, small molecules, or combinations of the two when you're looking at conjugate molecules. And especially in kind of relation to biological registration, the thing is that it's not directly transferable how you register small molecules over how you register biologics. In small molecule registration, you often know about your structure in advance. In biological registration, you often determine the structure of the biotherapeutic just in the very end of a very complicated forked decision workflow, which means you start off working with physical samples, and in the very end for your drug approval, you're actually going and looking at the entire structure and try to solve as much of the structure as possible. So this makes it very complicated because you have to track all of the individual steps during the development and still maintain, finally, the links which go back to the identity of your molecules, the identity of your conjugates. And this is kind of then located at the core, which I call the data relation management, stating that, okay, I know which cell lines I have used in the process registration workflow. I know which sorts of screens I have applied, which data came up in the assays, which sorts of criteria I have applied in order to select for the corresponding molecules, and then identify, okay, now I look at the structure and say, okay, this is my one and only unique structure, and maintain all of the kind of relational information attached to these entities. With respect to what goes in and what comes out of that system, we are aiming to provide something which is as flexible as possible in terms of the data sources which can provide data towards this registration system. This can come from our ELN, this can come from analytical data directly sped out by the LIMS systems, can be browser clients where a user just enters some sort of annotations or other feeders, and the business logic rules, which can be considered to be sort of exempt for each individual module. So there are kind of small sets of rules which describe how individual macromolecule structures are registered. But business logic also means that if in one organization the workflow considers the source, for example, how a biotherapeutic was developed, and another one says, I don't care whether it's produced in different cell lines or not, then we'd like to keep this configurable in a flexible fashion here. And then further downstream, you'd look at somehow an ability to query your data in order to kind of push out the information registered in given IDs and link corresponding data to that individual entities, and then kind of create data marts on which you can do your SAR analysis, your data analysis, and have further kind of downstream applications which consume the data based on individual IDs given in these registration systems. Now, maybe a small illustration with respect to how that could look like in terms of this relation management. Look at a candidate clone for monoclonal antibody production. Then in the end, what you'd like to do is you'd like to identify the activity or specificity against a specific biological target. And well, that's not really dealing with any sort of small molecule registration. So this is just disabled for this case. Macromolecule registration also doesn't really apply simply because, well, you just have a candidate clone with a given activity. You may not know the sequence of it yet. So what is really interesting at this stage are these two things down here. You may have a physical sample, you may know the cell line which is produced, and you may know how you have created this until now. The kind of information management dealing with setting up these relations is encoded in a rules layer, which in the end kind of brings certain things together in a very ordered fashion. So if you're looking at an antibody that can be produced by a cell line, the cell line itself is registered in the biomaterial registration. The antibody, once you know about the sequence, would be registered in the macromolecule registration module that can inherit already something which is just a different entity, if you like, in the macromolecule registration space, which itself is a general peptide, and certain parameters or descriptors of a peptide are automatically inherited by the entity of an antibody because, well, the antibody has peptide sequences, gene names, descriptors, and things like that. But let's go to, this is more like a future outlook to what we are aiming to develop. Let's go back to what we actually have developed so far. So you've heard from Akos that we have it looks rather complex small molecule registration system, but in fact it can be configured in a way that most of these things do not fall to any sort of staging area with manual intervention. So they can be set up that it's in fact an automated small molecule compound registration system. And what we've recently been doing is we have been developing a registration module for the structure of macromolecules, which is based on HELM notation. I'll just spend a few words on this. So summarizing, and biological registration challenges are in fact threefold. One of them is the structural component. There's something related to what sorts of additional metadata do I have for the individual entities. And finally, how do I treat the interconnectivity in terms of workflows in which I'm developing these biologics. For the first one, the major challenges are that we are currently still lacking proper representation standards for the atomic structures of such molecules. And how do we input these things as a scientist? How do I kind of push this information into an information management system? We've seen it in Marvin. Obviously, that's not the right thing to do, right? On the annotation side, you have lots of closely related things. However, they come with very distinct descriptors, which if you want to encode this in a relational database, for example, you would end up with a very sparse data matrix. Some of the entities have only a few parameters, but all of them are very different in themselves. And then the additional challenge is that depending on the domain you're working in, you may end up with different names for the same thing. So medicinal chemists may come up with a different vocabulary than your biologists. Still, they are talking about the same thing. You got to handle this somehow. On the business logic side, the uniqueness of biologics often is dependent on the genealogy. I mentioned it a couple of times. And the second problem, though, is even worse, in fact. The process workflows are often changing with new technology evolving from academic research and basic research. There was a neat example about this. I've been talking about a company about antibody registration. They said they've spec'd out with a consulting company a kind of defined workflow on how to register their antibodies. This process took about 14 months, and then they implemented the system in another 2 months. And unfortunately, 6 weeks later, they've had a major revision of their antibody development workflow, which rendered the entire system essentially useless. And obviously, we are aware of it, and we need to accommodate these kind of technology changes while being able to provide a system which is flexible enough to cope with that. So in our long-term goals, how can we tackle these individual problems? Regarding the representation, we've decided to take a look at the HELM representation, and we think it's a viable way to describe large molecules. Input methods, obviously you need some sort of a new tool, a biosketcher, which enables handling this sort of new representation and embedding, whenever chemistry becomes important, the classical chemical sketchers. Regarding descriptors, if you come up with an agnostic data model, which doesn't care about what the entities in fact are, but kind of encode the entities in a rules layer, then you may be flexible enough to end up with a descriptor system or a descriptor management system, which allows exactly that. Domain dependence annotations essentially are being addressed by the use of ontologies, which in the easiest case can come up to something like simple synonym tables. On the business logic side, like the integrated registration approach, which I've shown to you in the initial slide, is essentially what we imagine would be able to address these challenges. And on the side of the highly dynamic workflow, there is the process registration module. In the short term, like, okay, what do we do right now? We are addressing these three. And what we do is we call this our biomolecule toolkit. And this is the stage at which we are. And now I'd like to tell you a bit about the biomolecule toolkit. So we have something which has been externalized from Pfizer. So they've promoted this from within the Pistoia Alliance as a representation standard for large molecules. And we as ChemAxon, we have invested a lot of effort in order to externalize their tools and make them available as open source tools and put this to the public domain. Both of these tools, so a HELM notation toolkit, essentially bundling all functions relevant for handling the representation layer, and then the HELM editor, which essentially is just a graphical user interface working on this notation. Both come with an API. And we have decided to build a kind of registration web service layer around this HELM notation toolkit. We've also put up I call it demo application. Essentially, it's like our testing site where we can test all of our services for functionality. But we are soon looking at integrating this with actually our regular registration system. And at the same time, we have integrated some helper service down here, which allows us to kind of manage the data, which is required by the HELM notation language with our kind of web service layer and the graphical user interface. But I go into the details about this a bit later. From this toolkit, we communicate with our web service core at the moment with a simple HELM notation string, which contains all chemical structure information of a molecule. However, we have a set of services which allows you to kind of come in with a variety of things like sequences. If your peptide or nucleic acid sequence, you can come in with FASTA files or a regular HELM notation string. We are looking at extending this also to regular mol files or SMILES. And the only thing we have to do here, probably with the help of standardizer, is we can do an automated extraction of the kind of biological context of a molecule. If you had S groups defined superatom groups, sometimes they are stored. If you're running this in MolV 3000, then we could readily extract this sort of polymeric information and just run it right away. If you don't, then we need a sort of parser in order to extract the sequence from the incoming SMILES, for example. But that's future work, why it's this entire web service core is something which we have right now. And it deals with format conversions, standardization, and we also put some effort into canonicalizing these HELM notation strings so that we can actually register them based on this HELM notation, and then put up a number of DEO services in order to kind of register a molecule in a database, register all of the relevant monomer information on the HELM notation side. So I think I've mentioned this, so let's just move on forward. What I'd like to point out is that if you haven't heard about HELM before, and I think it's somehow lingering around in the R&D community for quite a while already, there is a project homepage kind of maintained by the Pistoia Alliance, openhelm.org. It's a really comprehensive resource where we have put up tutorials for the usage of the editor, where we've put up just very recently the kind of first version standard specification, and all of further activities will be probably listed there. In fact, I gave just last Friday a webinar on technicalities of the HELM notation. I think it will be linked in a YouTube video here, so if you're interested, just browse the page to see what HELM actually is and how it can be applicable for you. This is also HELM. That's the HELM notation of the antibody I've shown you in the first picture, in fact. I don't want to go into the details, but as you can see, it comes with a lot of symbols. It is complete, so the entire string is the HELM notation of that molecule. The other thing which is missing is that the individual symbols you're using in here are not defined in the output of this HELM notation, but I will go into that a little later. So they actually contain chemical structures with their attachment points, certain rules about how they interconnect, so they are needed in order to interpret such a notation string. And we used to say that this sort of HELM notation for macromolecules is essentially like the SMILES which you encounter for management of small molecules. So it's, if you're experienced a little bit, you can read it easily. It comes in different sections, which I've colored here, about the individual linear polymers contained in the molecule, how they are interconnected here in the blue section. And you can even put up some annotations back there stating, okay, which ones are the light chains, which ones are the heavy chains of my antibody. So that's the current stage. And this is just a list of services which are available. You find this in the presentation, which is on your pen drive, so I don't want to go into details here for time reasons. where do we go from there? I've said that one of the most urgent things we are looking at is the integration of a small molecule registration system, which we have, for the reason that one of the frequently asked for use cases is, do you have a tool which handles not only biological macromolecules, but also conjugate molecules? And conjugate molecules often come with additional complications, like you have your antibody-drug conjugate. The conjugation reactions is somewhat statistical. So you essentially have a conjugation pattern around it. That's something which the HELM format at the moment doesn't support yet, but I think that's pretty high on the priority list in order to enable this. And we see good chances in order to define such extensions not too far in the future. And then you can imagine further calculators, kind of blast services for searching the content you have stored in the registry. You may want to integrate information which comes from your crystallography and experiments. When you've got a PDB file, you want to do a mapping of the kind of individual atom coordinates to the actual atoms in the chemical structure, for example, in order to do the further real structure activity relation analysis. I could go into details about the example here, but I think the main message is that we can do this sort of integration with the compound registration from both sides. So starting off from the registration of HELM, what is required is that if you had the example of such a antibody-drug conjugate, is that the antibody is somehow represented in your string. The corresponding linker is a peptide linker, which is indicated here. And you have the corresponding kind of annotation for it that can come with a compound identification number from your registration system. So you can at any time look it up in your compound registration. And the conjugate itself is kind of coming either by SMILES, also via a compound identification number. And then you have some further information down here about, okay, in which ratio is it bound to the antibody? What's the ID of it? And you could kind of enrich this notation with, well, in the compound registration, we call it chemically significant text. So there are additional descriptors which are related in order to fully describe the molecule you're looking at. But obviously, you can also do it from the other side round. You have things registered in your compound registration. You want to pull this over to your biological registration system because you have identified a number of compounds which may make suitable warheads for your ADCs. Then you already have these sorts of compounds. The only thing which you need to do in order to use them in HELM is you need to define an attachment point, like how is the connectivity of this if you're using a specified conjugation reaction. There can come up a pop-up window, as you have seen it for Marvin choosing free attachment point. So you choose the atom, which is the final attachment point, and specify what is the leaving group of this, and then essentially you have pulled it over into your HELM molecule registration system. Okay, so what do we have now? We have an evaluation version ready at the moment. We, in fact, have implemented a small demo site on ChemAxon.com. If you're interested using this, then please contact me, since we have put a little password authentication. I would like to really encourage you to talk with me because I want to know what you're looking for. And the toolkit comprises of the database with some auxiliary services. We actually support both SOAP and RESTful web services. So it's no issue switching between the two of them. And afterwards, these kind of services are brought together in a single demo page where all of the kind of functionalities we've bundled with the toolkit at the moment are exposed in buttons and workflows and things like that. So really, if you're interested, please come and talk with us. I leave this one here. I think we run out of time, but see for yourself.